Hello, everybody. Today, my team and I are going to present about genetically engineered insulin. Before I go right into it, I want to introduce myself. My name is Melva Rivera Chen, and my teammates are Alicia SJ, Natasha Christine Leong, and Ofelia Maureen Budianto. These are the core concepts that we are going to talk about today in this presentation. There are history and general information, structure, chemical bond, and bond stability, insulin synthesis, how to genetically engineer insulin, benefit, and ethical consideration. First off, we're going to dive deeper into the general information such as the definition of insulin. Insulin itself is a peptide hormone primarily produced by the beta cells in the pancreatic islets of Langerhans, consisting of 51 amino acids. It plays a key role in maintaining glucose balance, supporting cell growth, and managing metabolism. Working in conjunction with glucagon, insulin helps control blood sugar levels, promoting energy storage or anabolism, while glucagon supports energy release or catabolism. Insulin facilitates the uptake and storage of glucose in the liver, muscles, and also fat tissues, which can lead to increased body weight. Well, there is also synthesis in human body. Insulin is produced in the pancreas with the creation of pre-proinsulin, which is then produced into proinsulin. Proinsulin is then converted to insulin and C-peptide and stored in secretory granules awaiting their release on demand. Now we are going to discuss about the history, the pre-genetic engineering era. In 1889, von Mering and Minkowski found that dogs without pancreas developed diabetes, the first link between pancreas and the disease. In 1921, Banting and Best extracted insulin from the ligated dog pancreas, a major breakthrough. In 1922, Leonard Thompson became the first human to receive insulin, and insulin was extracted from animal pancreas such as pigs or cows, but it caused immune reactions and inconsistent effects. In 1936 to 1946, Hans H. developed longer-acting insulin using protamine, leading to NPH insulins. In 1950s, animal insulins were found to cause anti-insulin antibodies and lipodystrophy, prompting the need for purer forms. In 1955, Frederick Sanger determined insulin's amino acid sequence, marking the birth of proteomics. Now we're going to the post-genetic engineering era. From 1978 to 1979, scientists successfully cloned and expressed the human insulin gene in E. coli bacteria. In 1982, FDA approved Humulin, the first genetically engineered human insulin, safer and purer than the animal insulin. In 1996, the first rapid-acting insulin analog, or Lispro, was introduced by modifying amino acid positions for faster absorption. In the 2000s, insulin glargine, the first long-acting analog, was approved, offering a more stable, peakless insulin profile. In 2015, insulin degludec was introduced as the longest-acting insulin, up to 42 hours, thanks to advanced structural modifications. And today, we have a wide variety of recombinant insulin analogs. Rapid basal premix are available to match different patient needs with better safety and precision. And now we're going to talk about the insulin structure. We have primary insulin. It is the specific sequence of amino acids that make up two polypeptide chains, the A chain, which has 21 amino acids, and the B chain, that has 30 amino acids linked by the sulfide bonds. This is the primary structure of insulin protein. Chains A and B are connected by the sulfide bridges, which are covalent bonds between the sulfhydryl groups of cysteine residues. So these are the amino acids from the primary insulin structure. There are glycine, histidine, and glutamine.
Glycine is an amino acid that is part of the primary structure of insulin and plays an important role in insulin function, including blood glucose regulation and insulin sensitivity.
So histidine is an amino acid constructed by six carbon atoms, nine hydrogen atoms, three nitrogen atoms, and two oxygen atoms. So this amino acid is involved in the structure and storage of insulin, helping insulin stay stable before it is released to regulate blood sugar.
So glutamine is an amino acid that supports insulin secretion by helping pancreatic cells release insulin in response to glucose levels.
Now we have secondary insulin, which is the specific sequence of amino acids within its two polypeptide chains, the A chain and the B chain, linked by the sulfide bonds. The A and B chains of insulin can form alpha helix and the beta sheet structures, which contribute to the stability and function of insulin. Last but not least, we also have tertiary insulin, which is the three-dimensional folding of its polypeptide chains, stabilized by interactions like disulfide bonds and hydrophobic interactions between nonpolar amino acids, forming a compact functional sheet to bind insulin receptors and regulate blood sugar levels.
Okay, so there's three chemical bonds. In insulin, there's peptide bond, which helps assemble the polypeptide chain of insulin, disulfide bond, which maintains the shape and stability of insulin, and lastly, the hydrogen bond that helps preserve the structure of insulin.
The bond stability of peptide. A peptide bond is a covalent bond that is formed when two amino acids join together. It is created by releasing a water molecule during the bonding process with another amino acid. After the water is released, the peptide bond is formed. The bond stability of hydrogen. Hydrogen bonds are weaker than ionic or covalent bonds, but its bonds can also vary in strength, with some weak and some strong. The stability depends on multiple factors such as temperature, pressure, bond angle, and environment. Hydrogen bonds are crucial for many properties of water and other molecules. The bond stability of disulfide. Disulfide bonds play an important role in maintaining the function of insulin. Without disulfide bonds, insulin can undergo changes and may not function properly.
So next, I will be discussing about the process of insulin synthesis using recombinant technology. So first, we have to take the DNA containing human insulin. Specifically, we're using human insulin genes located in the chromosome 11, position 15.5 from human pancreatic cell. And then we have to get a plasmic factor. Usually, what's used is PBR 322 or PUC. And then we have to cut it open with a restriction enzyme. So we have to note that some of the DNA, and then DNA fragments and plasmids will have to be mixed. We have to note that some of the DNA fragments and plasmids will join together using DNA ligase and will result in what's called recombinant plasmids. While some others would not join together, and that is what we call non-recombinant plasmids. And then we have to mix the plasmids with some bacteria or yeast. Specifically here, the example we're using is using the bacteria E. coli. So in this step, usually what will be used is heat shock or electroporation. So the point of this treatment is to make the bacteria lower its guard so that the plasmids would be able to incorporate itself into the bacteria easier. We also have to remember that some of the plasmids will pick up the bacteria will pick up the plasmids, and this will result in what's called transformed bacteria, while some other would not pick up the plasmids. And then after mixing the plasmids with bacteria, we have to take the sample of the bacteria mixture on an agar plate containing the opposite substance of the resistant genes. So the transformed bacteria will grow. So this could be the recombinant plasmids or the non-recombinant plasmids. So for example, an agar plate with an ampicillin antibiotic will make sure that the ampicillin-resistant plasmids will survive. Next, we have to take some sample of both bacteria on an agar plate containing tetracycline antibiotic. So here, the non-recombinant bacteria will grow, while the recombinant bacteria would not grow because its tetracycline-resistant gene is rendered non-functional when the inserted DNA fragment was inserted. So now we can differentiate between the non-recombinant bacteria and the recombinant one. And then we have to take the recombinant bacteria and put it in a fermenter or what's also called bioreactor. In this stage, the plasmids will then multiply in the bacteria, and the bacteria will then divide as well, producing more insulin genes. It is important to note that all of these processes are done separately between the A amino acid chains and the B amino acid chains. So then we can extract the A and B chains individually and separately. Okay, so next, the final steps. So insulin A and B chains are purified and mixed under oxidizing conditions and then folded to mimic natural human insulin structure. So in this process, three disulfide bonds would be formed. So two interchain between that is the disulfide bonds formed between chain A and chain B of the insulin structure, and then it would also form one interchain disulfide bond, which is the bond formed within chain A. And then, in order to purify this proinsulin, there are three methods to do it. So, one is chromatography, two filtration, and three dialysis. Lastly, I have to mention that peptide bonds are different from disulfide bonds. So peptide bonds are covalent bonds formed naturally during translation in the ribosome in the host cells. So the bacteria in this case is the E. coli, and this bond links each amino acid with another amino acid. So different from peptide bonds, disulfide bonds are formed between the two amino acid chains and are formed by covalent sulfur bonds. So remember that disulfide bonds bonds between amino acid chains, whereas peptide bonds links between each amino acid. So it links between amino acids forming polypeptide chains. Next, I will discuss about the benefits of the recombinant technology. So first of all, it is safer because the quality and cleanliness of the insulin hormone can be controlled as it is produced artificially and clinically. And then it will be more widely available as it is also mass produced by using artificial methods, which cause it to have increased accessibility to us. And then it would also have more variants because it is artificially made. Therefore, producing insulin opens door by modifying it to suit our specific needs. And lastly, it would also be more affordable because the ability to mass produce insulin within a short time makes it more efficient and cost effective.
Ethical consideration of genetically engineered insulin. So, pharmaceutical companies have exploited the patent system and market exclusively to have less competition, which would grant them to charge customers with higher prices. Some companies increased the prices of insulin on 13 occasions. And not only that, the prices of some insulin has increased by around 17 times over 10 years. The control that the pharmaceutical companies hold from patents give them power on increasing insulin prices. It is evident on how many times insulin prices has increased in these past few years. This action would have a huge impact on people's lives because less and less people could actually afford to get insulin.
So this is the sources that we used. Thank you for listening.